I've also tried to prepare the talk thinking of about two facts. One is that I believe that your team is mainly interested in early diagnosis screening, and that you have a lot of interest in liquid biopsy. I'm not going to deal with any of that, but I think some of the slides that I have chosen to present have been chosen precisely to have you think about why pancreas cancer is a tough business. Okay, so I'm going to talk a little bit about pancreas because it's important to know what is the scenario where we move, about the genetic growth to pancreas, a little bit about pancreas subtypes, and about genetic mouse models. And my principle about doing the lab is that you do in the mouse what you cannot do in human. What you can do in human, you do in human, because that's the perfect model, that's reality, okay? But I'll tell you a little bit how the models get help. So this is the pancreas. What you can see here, for those who are not familiarized, in blue is the ductal system. Anyway, what you see in red or yellow are the islets, and everything that's dark is the real stuff, which is the acinar cells, the exocrine pancreas, the real stuff for those who are interested in exocrine pancreas cancer. Yuval makes fun of me, which is right, because we know that we like to talk to the endocrine as well. And what you can see here in this slide is an H&E section of the pancreas, where you can see that actually acinar cells are the most abundant cells in the pancreas. And that's important because for a long, long time, we thought that the most common pancreatic cancer, which is pancreatic ductal adenocarcinoma, arose from pancreatic ducts. And now all we know is that we don't know, because we know that in the mouse models, ductal adenocarcinoma can come from ductal as well as from acinar cells. And acinar cells are much more abundant than ductal cells, that you can see here. So what's the scenario? The scenario is that, and you know about this, but the numbers are inspiring. If you diagnose a 2-centimeter tumor in a patient with breast or colon cancer, 5-year survival is 90 to 95%. If you diagnose a 2-centimeter tumor in a patient with lung or kidney cancer, two typically bad tumors, 5-year survival is 60 to 80%. If you diagnose pancreatic cancer of 2 centimeters, 2 years, not 5 years, 2 years survival is 20%. So the message is that in pancreatic cancer, early is too late. And that's your business. Early is too late. We're interested in understanding what happens, at least my lab is interested in understanding what happens very, very, very early, because early is too late. So this is the pancreas again, and let me tell you a little bit about the pancreas, because it's important, I think, in order to understand possibly some of the features of this tumor and some of the challenges ahead. Acinar cells, which you can see here as these pinkish cells here, which you can see here, ultrastructural analysis. Acinar cells are professional factories for making proteins, and they make huge amounts of proteins. Their specialty is making huge amounts of protein. You can see the rough endoplasmic reticulum in the panel on the right. And actually, these 25 genes, which are all made by acinar cells, except for maybe I removed insulin here because it's not an acinar gene, these 25 genes account for 80% of all mRNAs in the pancreas. So 25 genes accounting for 80% of the mRNAs doesn't occur in any other tissue. So they are professional producers of these proteins. And not only that, I have to emphasize that all the genes that you see here that code for proteins that are labeled in black are enzymes that can hydrolyze DNA, RNA, protein, carbohydrates, etc. So if these enzymes leak into the surrounding tissue, they can cause important damage. And this is one of the causes of pancreatitis. And we'll talk about pancreatitis. There's no other tissue like that. It's made to disrupt what you eat. Okay, that's the message. Important things. That's the normal tissue. This is pancreatic cancer. And pancreatic ductal adenocarcinoma, and Dan is here and knows more about this than I know, has a very rich trauma. You can see here, I cannot show you, but you can see here, these are the tumor cells. These are the tumor cells, but the vast majority of cells in what you see here are stromal cells. And this has important implications, both in terms of understanding the pathophysiology of the tumor, but also possibly in terms of understanding leakage and of DNA produced or shed by tumor cells or extracellular vesicles or whatever from the tumor cells towards tumor glands to the circulation. Okay, so if we are interested in understanding, you are interested in understanding early diagnosis and screening, you have to know what the causes of pancreatic cancer are, and this is a summary that we prepared together with Noria's group. And you can see here in red all the risk factors, in green some possibly protective factors, at least there is an association at the epidemiological level. And what's common to many of these factors is that there's a strong inflammatory component, possibly related to what I told you about pancreatitis. So what do we know about the genomic, how pancreatic cancer develops? This is, there are 3 roads to pancreatic cancer, as far as the pathologists can tell, and this is verified by molecular methods. We know that there is a road that, whether it starts in normal ducts or possibly in acinar cells, from what we know in the mouse, we don't have any certainty in humans, that goes through PANCAIDs. PANCAIDs are pancreatic intraneoplastic neoplasia, low grade, high grade. The ones A and B are now called low grade, the 3 is called high grade, and then it's PDAC, IPMNs, and mucinocystic tumor. These are a different route, probably accounting for about 15% of the cases of pancreatic cancer. What do we know about the panning role, which is the most important? Well, this is what textbooks say, and I don't think it's true, but I'm going to show you just because the textbooks say it, okay? So you know that I read the text. What is known is, and this has been extrapolated from the study of pancreatic cancers, and then, so what you can see here has been learned backwards from studying PDAC samples, and what we know is that in PDAC, 90% of tumors contain KRAS mutations, 100% of the tumors harbor P16 INFORT-A inactivation through one mechanism or another, and 90 or let's say 90% of the tumors contain alterations in the P53 pathway and the TGF-beta receptor pathway. So first message I want to say, this is a genetically very homogeneous tumor. It seems that alterations in these 4 genes are a sine qua condition for the development of pancreatic cancer. I cannot think of any other tumors in humans that behaves exactly this way. And what is known is that what has been done is in patients with PDAC, largely in patients with PDAC, and I'll go back to that later, preneoplastic lesions have been found, or putative preneoplastic lesions have been found. Pathologists have known this for years. They gave them this name. And we know that in low-grade findings, we can already detect alterations in KRAS. I'll go back to that in a minute. And in high-grade lesions, which are thought to be precursors to PDAC, we have alterations in P53 and in SMART. Okay, let's go a little bit into RAS, because obviously, for somebody who's interested in liquid biopsy, detecting KRAS mutations, possibly in pancreatic cancer, appears like a very promising strategy, because 90% of tumors are KRAS mutant. So mutations are found only in KRAS, very frequent, also found in panels, can be detected in a variety of biological samples, juice, pancreatic juice, stool, cell-free DNA, studies going back to the late 90s. Mutant wild-type allele imbalance, I'll go back into that a little bit later, are associated with tumor progression. So an oncogene where there are allelic imbalances. And you can see here actually that these allelic imbalances may be very important because patients whose tumors show an allelic imbalance have worse outcome. Second important message. What do we know about all the genes participating in this tumor? Well, this is from the first paper in Nature from the ICGC, Australian ICGC, by Bianchi et al. 100 tumors. Only 4 genes were mutated in more than 6 of those 100 tumors. 1 gene was mutated in 6. But what you can see here, therefore, and these 4 genes that were mutated in more than 6 tumors were exactly the 4 that I've told you before that had been identified through candidate or standard non-NGS based technologies. So that means that the majority of pancreatic cancers have mutations in what I call the magnificent 4, plus a myriad of private mutations that we don't know which of those are truly relevant to the tumor or not. And it's difficult to sort that out because they are infrequent. Well, that was 100 tumors, but this is foundation medicine, clinical samples, real world, whole exome sequencing of 16,000 pancreatic cancers. Top 4, KRAS, P53, P16, SMART4. All the others, private, low frequency mutations. So this is challenging. because we have a big player, KRAS, and then the others are much less. Well, is that true? The group of Hopkins who developed the whole concept of the Vogelgram and who established the role of, not of KRAS, but yes, of P16, P53, and SMAD4, actually did a very interesting and important exercise that people tend to forget, which is they went back and said, well, let's study lesions, pre-neoplastic lesions from individuals without pancreatic cancer. And of course, if they are precursors, we should find them. It took an international collaboration of pathologists to find 23 individuals with a high-grade panning without pancreatic cancer. Does make sense, according to them. Well, it just means, I think, that panning 3 may progress to pancreatic cancer, which is a precursor lesion, may progress to invasive cancer extremely rapidly. And if you're interested in early diagnosis, that's important. But let's go into some more detail. They did whole exome sequencing in those 23 samples, and they did targeted resequencing in a few of them in order to look in greater depth. And what they found, these are high grade. This is panning 3, which according to the slide that I showed you here, has mutations in P53 and SMAD4. Well, sorry, has mutations. So look at this. It turns out that high-grade lesions have mutations in KRAS, most of them. A fraction of them have mutations in P16, but very rarely mutations in P53 and SMAD4, which leads me to think that the model we have is based on the analysis of the wrong samples. It's based on the analysis of what looks like panning 3 from patients with pancreatic cancer, which in fact is intraductal infiltration and field camperization within the ducts. If you're interested in early diagnosis, this is important. Nobody pays attention to this paper, by the way. We keep publishing the same Vogelgram all the time. But the people in Hopkins are very good, and they have made very important contributions to the field. So they went back. And what they did, this is a paper in bioRxiv, not published yet. It will be an important paper when it's published from last year. So what they did is they took panning lesions from a number of individuals. They did very extensive 3D reconstruction of panning lesions from a number of individuals with pancreatic ductal adenocarcinoma and some with other tumors, but non-pancreatic cancer, non-pancreatic ductal adenocarcinoma related. So neuroendocrine tumors, for example. And they did 3D reconstructions, and some interesting things emerged from this very exhaustive and very important analysis. First, they could map that in one single individual, there could be many panning lesions, which is not unexpected. Matthias has started this in the 90s and has published on this. And you can see here each individual panning labeled in a different color, 3D reconstructed through very exhaustive sectioning of the pancreas and 3D reconstruction computational. And then this is a summary. They were estimated that there are 13 pannings per cubic centimeter in these individuals, a selected group of individuals. These are not known. They extrapolated that there may be up to 1,000 pannings per pancreas. They found that each panning contains from 27 to 7 million cells. So pannings is a world, very diverse. All pannings had KRAS mutations, but no other gene was mutated in these pannings. They did whole exome sequencing in more than 20% of the pannings. The average number of mutations was 13, and in some pannings, more than one KRAS mutation could be verified, indicating that they may be polyclonal. This is very important. This is really fundamental if one thinks of what we're going to learn from this in order to plan the screening or early diagnosis. Okay, and then Maria Pasca Di Magliano published late last year in Cancer Discovery, another very important study, which is based on the analysis of samples from normal individuals, organ donors. So these people don't have any known disease in the pancreas, and they did very careful optimization of methods to collect the cells, to analyze the presence of pannings, and to assess the whole pancreas through regioning the head, the body, and the tail. And what they could say is that they could find panning lesions in all individuals, regardless of age. Of course, the frequency was higher, as Matthias had published a few years ago, and other people have done the frequency of panels is higher in older people than in younger people, as one might expect. But the important thing, and I'm not summarizing this paper because it's published and you can go and look at it, is that they show that the panels from these individuals are different from the panels from individuals with pancreatic cancer. And they can find that actually there are differences, particularly in the microenvironment, in terms of macrophages and normal fibers or non-neoplastic. Two very important papers that we have to reflect upon in order to understand what's going on in early stages of tumor injuries. That's mutations. We know that mutations, or the current evidence strongly supports that mutations probably occur early and copy number alterations occur later. This is taken again from one of the Australian ACGC papers in Nature, 2015, looking at copy number alterations. And I will come back to this because this was done on macro dissected tissue. And what you can see is that indeed there are a number, obviously you can imagine that detecting or identifying copy number alterations in a tumor that is so desmoplastic is a challenge, because sometimes only 10 to 20% of the cells in the tumor sample are actually tumor cells. So this has to be taken with a grain of salt. About 20% of the patients, of the tumors, were relatively stable. About 30% was very highly rearranged. Some of them were considered to be very highly unstable, associated in association often with DNA repair. So very marked alterations in copy number changes in pancreatic cancer. And this is, to me, the most important paper published in the last decade in pancreatic cancer, genomics of pancreatic cancer, from Fayaz Nota at the OICR in Toronto, where what they did is they did laser microdissection of the cancers so that they could assess not only copy number changes, but they could look at allelic imbalance, they could look at specific mutations, and what they found are two very important things. One, they could find evidence of haploid event, whole genome duplication, in a very high fraction, more than 70% of pancreatic cancers. And then because they had microdissected material, they could do a careful mathematical analysis, looking at whether the mutations and the copy number changes had taken place before or after the whole genome duplications. And what you can see here is that mutations, either age-related or double-strand break associated mutations, were very similar before or after the diploid event, strongly suggesting that they had occurred earlier. By contrast, copy number alterations occur after the diploid event. So what we think now is that many or most of the mutations that are involved in pancreatic cancer occur relatively early. Then there's a genome event duplication, we don't know why. And after the genome event duplication, structural abnormalities take place. So he was able, because, or his team was able, because they had done laser microdissection, to do very careful analysis of copy number alteration. And they described that in at least 50% of the tumors, they could find evidence of this process, which many of you probably are familiar with, chromothripsis. For those who don't know, chromothripsis is can be explained through shattering of chromosomes, and then the cell tries to fix it as it can using non-homologous recombination repair. And what he was able to show, and this is very important, again, for those who are interested in how pancreatic cancer evolves, is that in some of these tumors, multiple chromothriptic events took place at the same time in the same tumor in different chromosomes. And he could show that in at least 20% of the patients, there were multiple chromothriptic events affecting multiple chromosomes, and some of the chromosomes involved are chromosome 9, where there is P16, chromosome 17, where there is P53, and chromosome 18. And he could map that in a proportion of these tumors, alterations, simultaneous alterations in these three genes, and in some cases only in two, had occurred, suggesting that there is a genomic catastrophe that could herald why there is no PANCAID in the normal population, because there is very rapid evolution from a pre-neoplastic lesion to an invasive tumor. And this is essential if we want to understand how to apply early screening or how to understand the early diagnosis. So this goes back to a hypothesis that humbly I had proposed 20 years ago and nobody paid any attention in 2013, which is a catastrophic progression model for pancreatic cancer, which suggests that as in ecology, this is driven from ecological studies, instead of being a gradual process, the development of pancreatic cancer, in many patients, not in all necessarily, but in many patients, it occurs as punctuated equilibrium in ecology. And what it means is that we may have KRAS mutations that occur early on, and maybe they are around, maybe no, they are around in many KRAS mutated panning lesions in probably most of us, most likely all of us who are older than 50. And then there may be a catastrophic event that involves one or more of these genes or these loci that are essential for a very rapid progression. And this, to me, is very important. What do we know after that? Well, what we know is that once this has happened, things are relatively stable genomically. This is work again from the Hopkins group, showing that actually, if you look at the metastases, we lack a lot of information on the metastatic makeup of pancreatic cancer because it's difficult to have multiple lesions. But again, the Hopkins group made a fundamental contribution in this regard. They could see that regarding point mutations, most of the mutations that were present in the metastatic lesions were already present in the primary tumor. Same happens in terms of copy number alterations, again suggesting that epigenetic events, or all the kinds of events that we don't understand, must be playing a fundamental role in progression to metastasis. This is the DNA genetics. Quick look into what these tumors look like. What do they look like? Transcriptomics, phenotypes, molecular phenotypes. Well, several papers, a number of papers, important papers have been published in the field in this last decade. The first one was by Eric Collison, where he first proposed the existence of classical and basal subtype tumors. This has become a standard in the field. We know that that's true. There are no basal cells that we know of, or maybe very, 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 very few, very hard to find in the normal pancreas, but the phenotype is common in tumors. We know that not only there are subtypes regarding the tumor cells themselves, but there are subtypes also regarding the stroma. Dan can tell you about that. And this has been verified in multiple studies. And what appears to be clear, because all studies concur on this, and there is no study that does not agree, is that patients with basal-like tumors do worse than patients with classical tumors. Okay, so this is important. Again, Ayaz Nora's group at the OICR in Toronto has made what I think is one of the, maybe not the second, but a very important contribution in trying to dissect what is the phenotypic diversity of these tumors by analyzing through laser capture microdissection the phenotype of the tumor cells. And he has refined a little bit the original proposal by Eric Collison and others, showing that there are possibly some subtypes of basal tumors, possibly also some subtypes. I say possibly because it has to be verified by independent series, but I think it's quite strong and the data is robust. Subtypes of classical tumors, and then a hybrid group in between, which can be trusted, the existence, because it was laser microdissected tumors. It's not just because of tumor dilution due to stromal contamination. One issue that comes up in the field is whether basal and classical is digital or is non-binary. Well, what this suggests is that it's analog. This is work from the Marseille group, showing that actually one can get scores of basality or classicality rather than a binary classification. And now we are in the 21st century, in the last decade of the 21st century, so we can analyze many of these questions, and this is important for pancreatic cancer because of the stromal richness that I told you. Now we can look at the tumor stromal cluster with single cell analysis, and this obviously is going to become very important. This is work again from the Norris group, which shows that in a small number of individuals who have been sampled at different times between diagnosis and progression with or without treatment, one can see both switching from hybrid stage to, in progression, basal-like phenotypes, or the opposite, from diagnosis basal-like phenotypes to classical phenotypes in progression. So plasticity can move the score of the phenotypes along, and we have to make sure that the way we define the phenotypes is proper when we compare diagnostic or pre-treatment samples with post-treatment samples, which is an important concept. This is essential. And of course, now we can go to spatial transcriptomics, and this is a paper from the MIT and the Broad Institute, again showing that what may matter is communities in terms of phenotypes and cell behavior, rather than simply each individual cell or only the tumor or fungus. Okay, so what I've told you is all human, and now I'm a mouse guy, so now I'm not a mouse guy only, I also do human, but we do a lot of mouse stuff, so I'm going to tell you why use mice. Well, we have all these factors that are risk factors associated with increased risk or decreased risk of pancreatic cancer. And of course, this is all studied when patients are diagnosed, or most of the data comes from studies where patients are diagnosed. So what the mouse allows us is to address questions as to which is the individual role of these genes, what is the role of alterations, because of course you will be surprised if I told you that some of these mutations might occur in the embryo. But it might occur in the embryo, because in the embryo, during those 9 months, you go from 1 cell to 3 kilos. So actually, everything important in your life happened while you were in your mother's womb. That's one thing that developmental biologists tend to say. The most important thing is how you gastrulate, not whom you marry, what you do in life, how you gastrulate. And we have shown, not in pancreatic cancer, but we have shown in other tumors, that some of the tumors that are diagnosed in the adult contain mutations that were present in the embryo. Okay, we have shown that in humans. So this can happen. We don't think about it. And of course, there are a number of environmental factors that may be affecting pancreatic cancer development, and we can study that properly. So, do we have pancreas cancer is the best tumor modeled in mice, in my opinion. We know that KRAS mutations, we can introduce mutations in KRAS, this is KRAS, this is P53, in P16, in SMAD4, we can introduce specific knock-in mutations, we can look at multiple KRAS alleles in the same context of P53 or SMAD4 alterations. We can do all of this, we can activate them in the embryo, we can activate them in the adult, we can try to understand what can happen in humans. We don't know whether it happens in humans, but what can happen in humans. So the models are beautiful. So I'm going to tell you a few examples of how we and others have used these mouse models. This is work from a summary work from Lofi and Munadodri at the NCI and from Nuria's group with some of the genetic variants associated with PDAC risk in humans. And that's going to call your attention. The top hit after ABO, which we don't understand what ABO does, but the top hit after ABO is NR5A2. It's a transcription factor that is involved in pancreatic differentiation, in acinar cell differentiation. So we thought that those variants, which are common variants in the population, would not be gain of function variants. Why? Because something that works, to make it work better, is very difficult. But to screw it up is quite easy. So we thought it would be partial loss of function. So we said, we're going to try to model this in the mice using NR5A2 heterozygous mice, which have loss of one allele in all cells in the organism, just like those variants. And when we looked at the pancreas of those mice, the pancreas is completely normal. But then, every time we find this in the lab, we do like cardiologists, what we call a pancreatic stress test. The stress test is pancreatitis. And what you see here is how a wild-type mouse recovers from induction of a mild pancreatitis. At 48 days, it's almost done. At 5 days, it's completely recovered. A heterozygous mice does not recover from it. More damage and poor recovery. This is the stress test. So now we know, and I'm not going to go into all the details, we published that, two things. One is that in the mouse, a germline variant, NR5A2 heterozygosity, cooperates with KRAS and with pancreatitis to cause pancreatic cancer in the mouse. And actually, many of these findings that we've made in the mouse, we've come back with Nuria to humans, and we've shown that these transcriptomic changes that we have seen in this histological normal pancreas actually can be identified in humans who have NR5A2 variants and who don't have any pancreatitis. So don't trust anything that looks normal, I'm going to tell you. Second, very beautiful example shown by Mariano Barbacid here and Carmen Guerra at CNIO. They looked at activation of KRAS and the when. When do we activate KRAS? In the mouse. If you activate KRAS in the mouse, okay, and you do it before birth, okay, before mouse birth, during the embryo, only KRAS, 10% of the mice die from pancreatic cancer. If you activate KRAS in the embryo and inactivate partially KRAS in the embryo, 100% of the mice develop pancreatic cancer. But this is in the embryo. Nobody believes, except for me and a few others, that something can happen during embryonic development. But now look at the following. This is activation of KRAS in the adult. And if you activate KRAS in the adult at 2 months, equivalence of age 20, then you get no tumors. No tumors whatsoever. Actually, the pancreas looks pretty much completely normal. The pancreas, I usually say, the pancreas cannot hear the music of RAS, the adult pancreas. But the embryonic pancreas can hear the music of RAS. However, if you now compare what happens when you activate in the embryo and you, in addition, cause a pancreatitis, you go from 10% to 100%. If you do it in the adult, no alterations in the pancreas. When you activate KRAS in the adult and you give a pancreatitis, you go to 30%. So the win is very important. And the other environmental factors, alcohol, lifestyle factors, whatever, can have a significant impact. So these are the things that we can learn from the mouse that can give us insight about how to look into the human data. This is just to tell you that mouse models allow us to get a further understanding of the complexity of other biological aspects of the pancreas that are important than is here. And this is just two papers published back to back 8 years ago, 10 years ago in Cancer Cell, showing that the stroma can either be good or bad. And that depending on how you manipulate the stroma can restrain pancreatic cancer growth, or deletion of the stroma, inactivation of fibroblasts, depletion of the stroma can accelerate pancreatic cancer. So these mouse models allow us to do this and allow us to answer fundamental questions on the biology. And finally, this is a very important paper from Ben Stanger's group, published in Cell 10 years ago, where they showed that actually in mice, high-grade panning lesions can shed, I'm not going to say tumor cells because they are pannings, pre-neoplastic cells into the bloodstream. So these can happen in the absence of invasive tumor. And actually they showed that with a number of genetic proofs, and they also showed that actually these cells that they isolated in blood, they do have recombined of the reporter that they had used, and they do contain the KRAS mutation that they had introduced. So things can happen even before an invasive tumor is diagnosed in a mouse. So if you're interested in early diagnosis and screening, this is interesting. And finally, mouse models are useful to study markers of disease. This is mass spectrometry metabolome analysis, and this is done in humans. And in humans, a number of alterations were found. This was done by Brian Wolpin and his collaborators at Harvard. And you can see that, cannot see because it's a quick slideshow, that actually some of the alterations that they found in individuals with pancreatic cancer in early, that may be precursors, that may occur in precursor stages, can also be found in the mice. This refers to branch stain amino acids. So in summary, what I want to say is that both the human and the careful study of the human and of the mouse data allow us to get some insight into the biology of this fascinating tumor, which is very complex, but that we need to take into consideration all these points if we want to understand how we can tackle issues of fundamental importance, such as the ones you're dealing with. And this is my group, and that's it.